So it's a talk, and we're going to see whether before lunch, the rise in urate levels we will have after lunch will be good or not for the brain. A good question. So again, thank you for this introduction. So I think that all of you are aware of this very old theory by which uric acid is good. It's good because it is thought to be a major antioxidant in humans. Uric acid, for the reason I told you before, have raised blood pressures and might have led to bipedalism. This is a very old theory. That being said, if you look at more recent papers, things are totally different because what we have learned from multiple research studies and from Tanya and Leo's lab is that uric acid might be not really antioxidant, but the opposite, pro-oxidant. As you can see, it can increase different signaling pathways, NF-kappa B, MAP kinase, it can increase activity of NADPH, leading to an increased production of ROS. And we do know that oxidative stress can be really deleterious for cardiovascular system and as well for the brain. So things appear to be quite complex because being antioxidant or pro-oxidant seems to depend on the chemical environment and seems to depend on the tissue we are talking about. So the most important is to have in mind the fact that there is really a clear correlation and a linear correlation between the level of urate in the blood and the level of urate within the cerebrospinal fluid, meaning that if you have high level of urate in the blood, you will have high level of urate within the brain. And what we know from a decade is that oxidative stress seems to play a key role in the pathogenesis of neurological disorders, and particularly in dementia, either Alzheimer or vascular dementia or mixed dementia. So it would imply that high level of urate seems not to be a good thing for the brain. If we look now at the overall mechanisms of uric acid, and if you read the paper written by Leo, you can see that, again, uric acid seems to be very deleterious when its levels is increased. As I said before, it can modify different signaling pathways, and particularly AKT pathways, can increase activity of the MAP kinase pathway. We have seen the impact of high level of urate on epigenetic, can activate the NLRP3 inflammasome, and we are going to see that the NLRP3 inflammasome might be involved in the genesis of Alzheimer's disease. So, as you said before, there is a strong correlation between inflammation and cardiovascular disease. And you know that there is a close correlation between vascular and the brain when we are talking about vascular dementia. You have demonstrated, you have showed the paper of the CONTOS trial. CONTOS was a randomized controlled trial in which patients were exposed to an IL-1 blocker, canakinumab versus a placebo. And in the CONTOS trial, the primary endpoint was major cardiovascular event. And it was clearly demonstrated that if you decrease the level of IL-1 in the blood of those patients, you can decrease the risk of incident major cardiovascular event. And a very similar observation was done in Colcott and Lodoco with low-dose colchicine. You know the story. When you prescribe low-dose colchicine at patients with coronary heart disease, you decrease by 30% the risk of having major cardiovascular event, showing you that inflammation is not very good for the brain, not very good for the cardiovascular system. If you look at this experiment, you can see that if you expose cells which are very close to macrophages, microglia, and if you expose them to amyloid beta, you can see that there is clearly an increase in the production and secretion of IL-1 beta, which is clearly dependent of the dose of amyloid beta. But if you block the NLRP3 inflammasome with a KO of NALP3 or ASC, you can see that you don't have this increase in IL-1 beta, showing you that these cells are clearly responsive to amyloid beta, are capable to produce IL-1 beta, so not very good for the brain, and clearly implicated in the genesis of the disease. So, do we have now data which would translate from what we have seen in vitro, in animal models, and in humans? So let's have a look now at all the studies conducted which have looked for an association between urate levels and an increased risk or not of developing dementia. The first study is the Rotterdam study. In the Rotterdam study, as you can see, a huge number of patients were followed up for more than 10 years, okay? And the authors have looked at a correlation between the level of urate, according to the quartile in this study, and the risk of developing dementia, adjusted, of course, in multivariate analysis for all the outcomes associated with an increased risk of developing the disease. And what you can see is that, according to the old theory, there is clearly a decreased risk of developing dementia when you have high level of urate, meaning uric acid might be indeed antioxidant and might protect against the risk of developing dementia. So when we produced the EULAR recommendation, we were really concerned with this story of uric acid levels, dementia, because with a drug, we can go very low. We can drop down the urate level to 2 to 3 milligrams. And we know from some case reports that going too low is not very good, might induce in some patients some edema in the brain. So we did a very similar study in France. And we used the 3CT Dijon study cohort. It's a cohort which is very well phenotyped because it was set up by the French government to search for risk factors for Alzheimer, because you know that the incidence and prevalence of Alzheimer is rising up. It's very costly. So the idea was to look for modifiable risk factors. So all the patients were very well phenotyped, MRI of the brain, and the diagnosis of dementia was done by an independent committee of neurologists. So in this, we did the same methodology, meaning we follow the patients at risk for Alzheimer, at risk for dementia, and we look whether the baseline urate levels correlated or not with an increased risk or not of dementia. And we did that without any a priori. We just have to look at the association of not. What we have observed is that, as expected, urate were clearly associated with GFRs, as I said before, associated with comorbidity, hypertension, cardiovascular disease, diuretic. It was expected. More surprisingly, what we observed is that clearly urate were associated with high CRP levels and high IL-6 levels. And low inflammatory grade is clearly a risk factor for dementia. So what was the results? What we observed is that in different models, and particularly in model 4, in which we adjusted for all risk factors, including the genetic, which is a key role in the risk of developing the disease, what we observed is that in the full model, we observed that there was a significant association between the highest quartile of urate level and an increased risk of developing all-cause dementia, Alzheimer's disease, and particularly in vascular dementia, as you can see here. So the conclusion of our study was that high level of urate is not very good for the brain for different mechanisms, which we can hypothesize, increase in oxidative stress, and so on. After that paper, a new study was published, this one, from, I think it is from Germany. They used again the same methodology, and surprisingly, they found the opposite of our result, meaning if you look at the baseline urate levels and the risk of developing either Alzheimer's disease, vascular dementia, or mixed dementia, they observed that the highest quartile was associated with a decreased risk. So conflicting results and opposite results as compared with our study. So it means that there are some bias. Clearly, epidemiological conventional approach, there are multiple biases. So if you are in this situation, the best thing you have to do is to look This is a brief summary of the studies I was talking about. You have to look at the Mendelian randomization study. You may know that this is a sort of statistical approach which measures variation in genes to assess the causal effect of an exposure, see urate levels, and an outcome, in our case, dementia. And it is very useful to assess the causality between two factors. In our case, urate levels, okay, and the risk or not to develop dementia. It has been done in many diseases, in myocardial infarction, to assess the association between LDL, for example, LDL levels, and the risk of myocardial infarction. And there was indeed a causality with this approach. What about MR, Mendelian randomization approach, and urate levels and dementia? This is the last paper, which is published in a very good journal, and they find nothing, no association, no signal, meaning in this study, which used 28 SNPs, okay, you separate the population according to a genetic score based on the SNPs, and you look at the prevalence of the disease according to the fact that you are predisposed to have high level of urate or not. By doing that, you randomize the two populations with the same environmental factor, same risk, same difference, high level of urate or no high level of urate. And there was absolutely no signal in terms of causality between urate levels, genetically determined urate levels, and dementia. If we look at other Mendelian randomization studies, it is absolutely the same results, meaning no association. So up to date, there is the level of evidence to support that urate levels might be causally associated with dementia is close to null. You are going to say, okay, but urate level is totally different from gout. Gout is high level of urate plus crystals. Do we see a signal? So let's have a look at the paper which would have looked at the association between gout and Alzheimer's disease. The first one is this one, conducted in Taiwan. Huge number of patients. You compare the incidence of dementia in gout patients as compared to control. You adjust for all the outcomes associated with the disease, and you look at the hazard ratio. As you can see here, there was no association. Excuse me, it doesn't work. There was no association if you look at untreated gout. No association between the disease and the risk increase or decrease of developing dementia. But if you take into account those patients who receive a treatment, in majority of cases XOI, okay, you can see that there is a dramatic decrease in the risk of developing the disease. Do we have another study? Yes. We have this one, which was published by a colleague in UK, sorry. And again, what they observe, same methodology, gout compared to a control group without the disease. You look at the impact, the association, and the risk of developing the disease. And again, what they observed is that in patients who receive a treatment for the disease, there is again a dramatic decrease in the risk of developing Alzheimer's. When you see that, you're not capable to say whether this decrease is due to the treatment, inhibition of the xanthine oxidase, because you know that the xanthine oxidase is capable to produce ROS in the vasculature system. So you might imagine that XOI might have an antioxidant property, might decrease the risk of developing the disease, or it is due to the disease in itself. Unfortunately, I don't have the response to that, because I'm going to show you that a paper has assessed the association between ULT and dementia, and again, the signal was null. So this is this paper. It was done in US, in Baltimore, by Yaz Singh. And as you can see, they found absolutely no interaction between having a treatment for the disease, XOI, and a modified risk of developing dementia. The last paper is this one. It was published some months ago. A huge number of patients in Korea, same methodology, gout patients, control group, you follow the patient for more than 10 years, and you look at the association between the disease and an increased risk or not of developing the disease, absolutely no signal. So it's very difficult to conclude. What we have seen from epidemiological studies is that one paper found a clear association between high level of urate and an increased risk of developing dementia, particularly vascular dementia, whereas 3 other studies found opposite results. If we focus on Mendelian randomization studies, 3 papers, no signal for a causality between urate level and a modified risk of developing the disease. And if we look at the disease conducted in gout patients, one study found an association and the other study found absolutely no thing. So it's very difficult to conclude. Up to today, I would say that there is no strong signal in favor of a modification of the risk. That being said, what we know is that uric acid might be considered as an LRP3 activator, so it might perhaps in genetically predisposed patients to do the disease might increase or accelerate the disease, but the level of evidence to support this theory is close to 0. So very sorry, I think that we can have a lunch without fearing to do something wrong for the brain. Thank you so much.